Well, thank you for the introduction. As mentioned, my background is in tumor immunology, so I came to CDI and to Seromics as a field from trying to monitor new responses to cancer vaccines, things that occur pre and post treatment. This talk's going to be a bit more generic as kind of an overview of what we're calling Seromics as a field. And so, what is Seromics? I think that's a good way to start this off. So, the human serum or Seromics studies the totality of the secreted antibody repertoire in serum, CSF, so other biologic fluids. So the most common and frequent place that you will find antibodies is obviously in the blood serum, but there are antibodies in pretty much every mucosal surface as well as other tissues. So just to kind of kick this off, so the human body is in many ways essentially a walking immune system, right? So about 30% of all of your nucleated cells are antibody producing B cells or antigen-specific T cells. About half of all the nucleated cells in total are immune cells. So obviously the majority of your body's tissue is not that because there's a very large difference in cell size. So the skeletal myocyte is much bigger than a B cell or a T cell. But the reason that there are so many unique immune cells is because the immune system essentially works on a random shape generation system. So every different little individual immune cell is kind of a dice roll opportunity at potentially binding some sort of insult, virus, or bacteria pathogen, pretty much everything that evolution on Earth can potentially send against us. And that unique section or those unique sections of the DNA, right? So the variable regions of antibodies, they encompass a really large search space, right? So there's potentially up to 100 billion unique B cell clones among those trillion or so unique B cells, and all of those clones are producing many times that of unique antibody molecules, right? And so if you're thinking about, the complexity of what you consider the human proteome. So you think about the genome, there's billions of nucleotides, it makes about 20,000 proteins, but those little variable regions in the antibodies, there's a lot of them, right? And there's actually way more unique nucleotide sequence in the B cell repertoire than actually exists in the genome itself. And the reason that there is such a vast search space is because, as mentioned before, these antibodies have to be able to defend against pretty much everything that evolution can throw at us, right? So pretty much all possible bacteria or viruses that have occurred since the origin of higher vertebrates hundreds of millions of years ago. Adaptive immunity has to be able to anticipate and predict that on an individual organism basis. And so all of this random shape generation is able to kind of help defend us. And most of the time we don't have to think about this because the immune system's there in the background doing its job. And a very important part of that job is trying to distinguish self from non-self. And a lot of ways and diseases, a lot of things that people are studying relate to where that gets misinterpreted, right? So when we think about what is Seromics, so Seromics is the study of exposure to things like viruses and bacteria and how good are our bodies at protecting from that. But where our work is really focused, and a lot of the clients that we work with, is where this kind of bleeds over and causes issues with the human proteome. So when cross-reactive responses to viruses unintentionally harm human proteins. And there's a really growing body of literature that it's the autoantibody serome that the real impact on human health tends to be found, and where there might be a lot of very interesting things to be discovered that our platforms provide access to. So as just one really clear example of this, before we get into the technology itself, so I'm going to claim that the serome controls human health, in particular the autoantibody serome. And what do I mean by that? So one really simple mechanism is you can have a good response against a pathogen, to a virus like Epstein-Barr virus, that then cross-reacts unintentionally with a specific piece of the human proteome, causing some sort of bad destructive result. And one very clear example of this that was published a couple of years ago in Nature is potentially the literal cause of about a quarter of all multiple sclerosis cases. And what they went in and did is they found that people that have had an Epstein-Barr virus infection, in some of these people, they will have a strong reaction to just this one epitope of a protein called EBNA1. And in some people, that cross-reacts with a protein called gliocam that's on the surface of human neurons, and that is potentially what leads to the immune system permanently destroying neurons chronically in myelin sheaths in multiple sclerosis. And so they've shown over here on the right, this is a mouse brain H&E stain where they essentially show that you can inject the mice with this EBNA1 peptide, and there's enough homology in the mice that alone is able to induce all of these immune infiltrates. And then they went back and kind of searched and were able to find that about a quarter of MS patients bear immunologic reactivity just to this epitope. And how did they find it? Well, they found it via unbiased whole proteome seromic profiling. They used an assay called VirScan, which we'll talk about a little bit later, that's got a bacterial phage-display library of pretty much all possible viruses as overlapping peptides. We're able to detect this epitope, and they found an isolated unique B cell clones from patients, and they used a tool called HuProt, or a human proteome microarray, to identify the cross-reactivity against gliocam, the human neuronal protein. And this target, right, this is really important. So the goal and purpose of the talk is talking about novel biomarkers and druggable targets from Seromics. Well, this is it. This is a landmark example because this peptide and reactions to it are a novel diagnostic biomarker for multiple sclerosis. And additionally, it's a novel druggable target, right? So you just have to induce tolerance to this one thing, and that entire complex cascading fractal condition that we call multiple sclerosis is potentially stopped by just tolerizing against this one peptide. And so by potentially developing a tolerogenic vaccine or a chimeric antigen T regulatory cell, you might be able to stop MS with just this one thing. And that was found through a Seromics assay. And so why am I the one here talking to you about this today? Well, it just so happens that our company, CDI Labs, produces the HuProt human proteome microarray that really the crux of that study was discovered with. And so I don't produce the proteins or make the slides. All the people on this slide here actually do. And it's really the culmination of quite a bit of effort to make this type of tool possible. So HuProt as a tool started about 15 years ago now. It was an initiative out of Johns Hopkins University, primarily with Hung Zhu, Seth Blackshaw, and Jeff Buca. Hung and Jeff are shown here on the slide, who got together with our co-founder, Ignacio Pino, to build the original HuProt microarray. So the thought was that by synthesizing individually the entirety of the human proteome, or most of it as actually occurred, you could use it as a cross-reactivity screening tool to see whether or not you were making high-quality monoclonal antibodies. So kind of a target identification tool, which actually was what was done here in this MS paper, where they used an isolated B cell clone combined with the microarray to figure out what was its target. But you can also do more complex assays as well. So the company was founded to build this microarray. And just to clarify that, we're a proteomics platform, but we make proteins and use them to detect things, either simple things like individual molecules or complex things like the serum, and to profile human autoantibodies. This is kind of the opposite of what other proteomics platforms do. So groups like Olink and SomaLogic, they detect proteins using different reagents. We make the proteins and use them to detect things like serum antibodies. And as kind of mentioned, there's kind of two different core things that a tool like HuProt is useful for. So you can imagine that having the entirety of the human proteome spotted on a glass slide. So as kind of talking about the slide, I should probably mention, so these are all GST tagged proteins synthetically made in yeast. So they're in a cell-based eukaryotic system, which really helps with the folding. So by having all of the protein degradation machinery in there, essentially we're only purifying proteins that have both that GST tag folded and that the yeast didn't shunt off to the proteasome before purification. And so we think we have a really good representation of full-length 3D folded human proteins that are then printed individually on the microarray. We can use this for detecting the response of individual molecules, deconvoluting unknown antibodies, or discovering novel protein-protein interactions. And we can also do whole proteome seromic profiling to look for autoantibodies in an unbiased way. So CDI as a company, as I mentioned, most of what we do is with the HuProt array. We also have a few other platforms. We have a virion display array of membrane proteins made in HSV1 or herpes viruses, where we can actually put a membrane protein in the viral capsid. We have most of the non-odorant GPCRs, as well as some immune checkpoint receptors. It's a much smaller collection than HuProt is at this time, so we're going to have the majority of the talk be about the human proteome microarray. We also have we're the first company to commercialize an assay called PhIP-Seq, or phage-display immunoprecipitation sequencing. This is a synthetic library of long peptides on the surface of T7 bacteriophages. And so you can chop up essentially the entire human proteome or the entire mouse proteome into overlapping peptides, right? And go ahead and actually use that to detect whether or not antibodies are captured in a pull-down assay. And then we do a sequencing readout. We also have a viral assay called VirScan. So it's sold by CDI Canada. And this is able to do a similar thing against all human viruses. And so how do we go about and actually present and deliver HuProt data? So the most thing is just really proving that the arrays are made successfully and the data and the assay is run in a clear and transparent manner. So we try to really be pretty open book about what the results are and how they are manipulated. And what's really nice about protein microarrays is if they're stained very well, the result is pretty clear. So this is an example of two different channels of HuProt data, and this is what things are supposed to look like. We have a foreground and a background of a human serum stained HuProt array. It's stained for a red channel, IgA, in green. And you can kind of see some little ghosts of things lighting up. We'll zoom in on that in a second. And that's what it looks like when you have an antibody against something. So we make the proteins once or twice a year right now. During those annual protein preps, we do quite a few spot checks to make sure that the proteins are made correctly. We then print them on a nitrocellulose membrane, and we stain with anti-GST to prove that all of those spots are down. And then within each individual array that is stained, we have spot pairs. So there's actually duplicates of every spot that's on a HuProt array, and we're able to verify that data is extracted similarly across the spots, which just kind of demonstrates that the data is clean and coming out correct, isn't generated by an artifact that's only on one spot or the other. And this is kind of what some outlier results look like. So this is a serum sample run on a HuProt microarray. And what these are just log z-scores within that individual array, or what are the brightest spots. And so these are the spots, or the pairs of spots that are actually coming out of this specific patient's array. We're seeing things that are relatives of P53 that are the top hits. This is actually a scleroderma patient that has anti-centromere B protein autoantibodies, and so that's coming up as a top thing. So that would have actually been a known clinical thing about this patient. We see that if you just stain with the secondary itself, so you take a separate array and you just do the anti-IgG, anti-IgA, we see that nothing's coming up, showing that these are real antibodies happening in the patient. And also on our QC stains for anti-GST on this individual array lot, we see that all of those individual spots are coming up GST positive. So we made a synthetic protein, it's there, it's not reacted to by the secondary, it is reacted to in the patient. So kind of these three things together tell us that these are real autoantibody signals. And so where we're really going with a lot of our analytics is we don't really subtract the background. We say, what's that foreground signal? You log transform it to kind of normalize the differences across the patients, whether or not they've got higher or lower levels of IgG. And we typically find that just looking for deviations within individual slides is enough to call real reactions. We also have, as mentioned, an entirely separate type of platform, a synthetic bacteriophage library. So you take an aliquot of these libraries, you react them with a complex serum sample, you pull down what gets captured, individually barcode and sequence the readout, and used mock immunoprecipitations without the presence of antibody to call real signals. So this is a very different type of platform, but again, accessing that same data type, right? So we're trying to figure out what types of antibodies do human beings have in a high throughput manner. And so similar to what I was showing with the HuProt reporting, we have a very detailed, thorough overview of, what are the QC parameters, how is the run go, how many reads did we get on each individual sample that we provide as part of the phage-display sequencing results. Just to kind of give you an idea of what the raw data looks like. So unlike HuProt, you're not seeing a literal spot light up, right? So what you're seeing are enrichments of counts in sequencing data. So this is an example of the raw data sheet that we actually provide. So we take the large FASTQ files and we summarize them for just how many times do we observe each peptide within the assay. So this is the VirScan assay, and so there's over 470,000 unique pieces of DNA that are representing all of these viral peptides. And these are different human serum samples. And what you'll see is that, the library is there in the background. Protein AG beads are innately sticky to the phage. And so you get a really good background profile of just that the library is there. And when you see a real reaction, it's going to be a really large deviation from that. So you'll get thousands of counts versus very low in the background. And I imagine these patients are looking a lot like mock IP if you actually ran the stats on them. So we provide the raw data, and then we'll call kind of individual hits based on the beads only background. And so kind of like with HuProt, we try to do minimal manipulations to the data. We give you all of those raw counts. And then we also go ahead and use a pipeline called EDGE-R, which is developed for mRNA sequencing to call significant reactions versus background distribution. And we can also provide case versus control outputs for different diseases. So these are actually some older individuals, people in their 50s, 60s, 70s, and above, with people that are teenagers or younger. And just this is one example of an outlier peptide to a specific herpes virus that it looks like none of the children have been exposed to, but quite a few of the older adults have been, and that other control samples are negative, that the beads only, so the mock immunoprecipitation without any antibody present is negative, and that the healthy young individuals look like that. And this is what you see when you see, kind of a real signal in the raw data itself. And what's great is because it is a VirScan, right? So this is an epitope scan of entire proteins. What you can actually see is it's a specific piece of this protein that is lighting up. And so now this heat map, so each column is a patient and each row is a peptide. And so it's, amino acids 49 through 110, 97 through 158, all down this peptide. And we have the old patients here, young patients here, and then 3 different control groups. We run these triplicate controls of either a pooled IVIG type plasma or a polyclonal against this protein called GFAP, which we use as a positive control. And this is now just a log fold change versus the beads only. And what you see is there's just this one specific peptide that's lighting up on this HSV2 peptide. And now here's another example. So earlier we were talking about Epstein-Barr virus, or EBNA1. So while we did not specifically run the VirScan assay for that paper, we are now running that library ourselves under license. And so this is now EBNA1 showing that quite a few of both the controls, so the control pool plasma, as well as the young and the old. So there's not differences between the cohort, but you see there's actually an overlapping peptide here where there's something in that region that's reactive in both patients, and that's reactive in both patients. And the peptides are not 100% overreacting, so you can get reactions like this that are only hitting one versus the other, but this is now identifying that the epitope's probably in that overlapping region. And so it's a really powerful thing that you could see this on this scale with the PhIP-Seq assays. And so, now we kicked off with one really clear example, did some overview of the human immune system. These are really the technologies that allow us access to this data type for the first time. But I'd like to kind of get into a little bit more about how is it that we think that the serome controls human health. And so there's quite a large repertoire of publications on this, over 200 now citing us specifically here at CDI, kind of that are helping create some momentum that Seromics as a field is kind of an underutilized data type. And so when you think about it, right, there's a lot of chronic diseases and there's a lot of money spent on what is the cause of Alzheimer's? What is the cause of Parkinson's? Why do some people get cancers whereas others do not? And the answer is at times there's some ghost of the answer in the genomics, but it's really clear that for a lot of chronic conditions, the answer isn't there. And the adaptive immune system is allowed to destroy cells on a very specific level. And I think there's a logic to the answer sometimes being in the seromic data. And as mentioned, we already showed the clear example of that in multiple sclerosis, but there's a lot of other ways that this can happen rather than just direct antibody killing. So there's a lot of information that's in the serome just as a surrogate of past exposure. So that's not always necessarily the mechanism that's there. So here's a really nice example of something kind of like that. So this is done by Iman Osman's group at NYU, and this is a validation cohort of hundreds of patients that have been treated with ipilimumab or nivolumab, so anti-PD-1, anti-CTLA-4, really big blockbuster checkpoint blockade immunotherapies. And what we've been able to show is that a pre-existing autoantibody signature, so before they're given treatment, there are things that these patients have autoantibodies to that predict whether or not they will respond or not respond, as well as whether or not they will have immune-related adverse events. And so there's probably quite a few things going on. It's a pretty large signature list of a couple hundred proteins. It's probably somewhat the immune system mirroring the type of tumor, but there's also some interesting effects in there, things like IL-2 receptor beta, that makes you wonder whether or not there's some sort of mechanistic thing underpinning that. And we'll kind of talk about that a little bit further into the talk. This is another kind of example demonstration of kind of patient stratification, now done with the VirScan assay. So we did this in collaboration with Benjamin Bessie's group at the Gustave Roussy, and essentially we're able to show in an unbiased way across all possible human viruses that infection with CMV or cytomegalovirus is very good in impairing anti-tumor immune responses. So you get fewer CD8 infiltrates into tumors, and people just don't in general respond as well in lung cancer when they've got an active CMV infection. This is something that's actually been known by tumor immunologists for quite some time, but to be able to show it in such a powerful way, in such a broad profiling aspect, is something that the VirScan assay enables, and something that I believe should be applied to quite a few more conditions. So one other mechanism that I think is perhaps the most interesting is another way that you can get drug-like effects and potentially do drug discovery with a different type of biomarker, which is unlike, say, the antipathogen response to a bad destructive autoantibody, where you can just have a direct killing effect. Antibodies can block immune pathways the same way that you can develop a checkpoint blockade therapy in a mouse that blocks PD-1 or CTLA-4. That things like that occur at some frequency in the natural human population. And Steve Holland at the National Institute of Allergy and Infectious Disease, I would encourage all of you to go watch this lecture that he did for an NIH webcast a few years ago. But he goes through and reviews in the infectious disease setting how there are certain types of chronic infections that people only get if they have naturally occurring blocking autoantibodies to really important things like interferon gamma, and that this just occurs at some frequency in the population, and that is what makes people susceptible to certain types of conditions. And one paper that actually went and showed this with HuProt microarrays was in a novel type of inborn errors of immunity that's similar to having an air knockout or APOSED, where patients have been shown in air for years that they'll develop anti-interferon autoimmunity, and that this anti-interferon autoimmunity affects whether or not they get certain types of additional conditions like type 1 diabetes. And they're just showing that this occurs in this other disease type using HuProt arrays. So you can get and you can observe using whole proteome tools like protein microarrays that you're getting all these anti-interferon or anti-cytokine autoantibodies that are affecting immune outcomes. That's really interesting. And we all know that interferons do things, and this is a really catastrophic autoimmune disease. And so these types of things occur and happen with a higher frequency. And so you can imagine that kind of in the infectious disease setting, you get an infection, and most of the time cytokines do their job. But if there's an antibody there blocking that, now you've got a chronic condition. And that's essentially what these examples on this slide are showing. And so kind of the thing that I'd like to posit is, so, cytokines are very interesting and we know they affect immune health, but there's a heck of a lot more surface proteins. There's a lot more things in circulation and on the surface of cells than cytokines. There's 3,500 surfaceome type proteins, and potentially all of them have some sort of condition that is occurring like this, or defect as part of natural human aging. So what else is being disrupted? And part of the reason I think that you should be so concerned about this is autoantibodies are a lot more common than you might realize. And so just to kind of give an overview of how many things like this each of us might be bearing. So let's go through kind of an overview of what is it that we know about the human autoantibody serome at this point. So the field is really still emerging. There's very few cradle to grave longitudinal studies. There's actually none that track a human being across the whole life course, but everyone, all of us, have autoantibodies, that those are very unique, that they could identify us at a crime scene, and that those would differentiate ourselves from our identical twins. And I'll show some data on that before I wrap up the talk. Most of the autoantibody serum likely arises earlier in life. It is stable across time, but additive. So things don't normally disappear. You keep acquiring additional immune memory for viruses and bacteria. That's a good thing. For autoantibody seromics, those are stacking mistakes that are occurring with age. And while not all of these autoantibodies are doing things, actually the general assumption is that most autoantibodies are doing nothing. They're binding an intracellular protein, or they're sticking on some non-functional part of a cytokine, and they don't really affect the outcomes. It's when things that are actually disease causing that we want to worry about those. So we'll just kind of run through some examples for each of these bullet points. So everyone has autoantibodies. I'm not going to show specific data on this, but just kind of take my word for it, having looked at thousands of these samples now. When you run an individual human on a HuProt microarray, there's going to be between 50 and 400 things that come up relatively high above background on that. So out of, about 1% of human proteins. 200, I think, out of the 21,000 is pretty common. It's the same on phage-Seq. So as mentioned, we have a whole human proteome phage-display sequencing assay, and this is a little bit more variable because it's a competition assay, but still, there will be a few thousand peptides that any one person, including myself, including you, that will have a reaction on these assays, even if we're not formally diagnosed with an autoimmune disease. It's just part of the human condition that there's going to be some level of background cross-reactivity that exists in the human serum. But there's not a lot of data on where this all arises. So Josh Laber's group has a nice paper using Napa arrays where they go through and profile people at different ages. And so these are not serial samples. These are just different bucketed age brackets. And they're just tallying how many antibodies they're claiming these people have at their thresholds at different ages. And the one thing that really stands out is the very young children have fewer than the kids that are, approaching puberty, and then that number stays pretty stable across time. And we've seen similar things as well. So how and where this arc of original autoantibodies comes from, probably from first viral infections and vaccines, where is the bulk of these mistakes that get made originally that you're then preserving across time? A lot of that's still to be established. And so I'd encourage any of you that have samples like that to come and do projects with us. So I'm claiming that the serum is stable, right? So they're in this paper claiming that it's pretty stable across time, but they're not doing serial samples, right? What you really want to do is look at the same people across a time gap. And so that's something that we did with a group out of NYU using our HuProt arrays. So you take serum, you react it with the protein microarray, you come back with a fluorescent secondary, you use a microarray scanner to see what lights up, and you get those pretty pictures like I showed earlier in the presentation. And so now this is just a little hierarchical clustering of lots of healthy women across a 1-year time gap. And it's a little bit hard to see, but what you see in the tree is you see all these little pairs that are coming up. And what you're seeing is essentially in an unbiased way in the log transformed HuProt data, the raw outputs of the microarray, everybody correlates with themselves. So everyone is more similar to themselves across a year time gap than anybody else in the study. And that's universal, and that looks the same across pretty much every sample that I've seen. Everyone is more likely to look like themselves than anyone else, and that this is a relatively stable thing across time. Now, these are all healthy people, right? So not a lot changes year to year. So your seromic profile is a relatively stable thing. Also, we tend to believe that the autoantibody serum is more unique than the antiviral serum. And the reason for that is it's the job of the immune system to see and protect us from viruses. There's lots of similar viral infections that are shared across the population, and that those are things that the immune system is supposed to do, right? So we all hope that we all have autoantibodies to the spike protein of SARS coronavirus 2019. So COVID-19 has unique epitopes, and whether you got it from natural infection or a vaccine, we are all having responses to those epitopes, but where we are having autoantibodies tends to be much more individualized. And so this is an example of a group that we did this with at Emory University, where early in the pandemic, we looked at we looked at children that either had COVID-19 infection or MIS-C syndrome after COVID-19 infection, or were healthy. And this is early in the pandemic, so none of the children have been vaccinated. And we ran phage-display sequencing on this population. And so now these are heat maps similar to what I was showing earlier. So each column is a patient, each row is a peptide. And this is pre-pandemic SARS coronavirus. And what we see is that the kids that have MIS-C or in the acute or convalescent phases of COVID-19 infection have these really nice, really strong cross-reactive autoantibodies that are binding the old pre-pandemic version of SARS. And that's the same for the nucleocapsid protein. You actually see more of the nucleocapsid popping up over time later in the infection or in the more severe state of MIS-C. It just kind of shows that, yes, there are similar epitopes that are shared across a population. Well, what if you zoom out and you look across the entire VirScan library, or you look across the entire human proteome? And so these are now plots of essentially that. So these are correlograms. So a bigger circle is a higher similarity of the PhIP-Seq hits between patients. And what we have over here on the right are the autoantibodies. And these are serial time points, the same patients at 2 different times, either during the acute or convalescent stage. The rest are all unrelated patients. And what you see is there's no filled circles out here in the rest of the field. And what that means is the autoantibodies that we're seeing in the HuScan Seromics are very, very unique, except between paired time points of the same individuals. Contrasting that to the viral side, so that the paired time points are still much more similar than any random crosspair, but you see you're getting bigger circles. And the reason is because across all possible different flu infections, your measles and your polio vaccine, there's stuff like this that's shared between people that's popping up all throughout the viral antiviral serum. And so that's more shared, so that the viral reactions are more commonly shared, and the autoimmunity is more unique. And so I'm also claiming that these responses accumulate with time and that it's more unique between identical twins. And so I'm going to show you probably some of my favorite unpublished data. We're hoping to get this out later this year. It's with Mario Falci at Twins UK. And what we've done is we've got pairs of lean and obese identical twins about 10 to 15 years apart. So these are all identical twins. Both twins are both lean, both obese. And we're looking at 2 different time points when they're about age 55 to age 70. And so we're looking for a few different things here. We're trying to see whether or not the autoantibody serum is similar across that large of a time gap, as well as are the twins sharing features of their seromic profiles. And so the first data that I'm going to show you is using HuScan Seromics. So looking pre-post across the whole human proteome, across this 10-year time gap with identical twin pairs. And what we see is that everybody looks like themselves a decade or more later, but they don't look like they're twins. So this is autoantibody data using PhIP-Seq. And what we have here are twin pairs. And so this is twin pair 8, initial time point, 8 after they've aged. Twin pair 8, individual 2, so the identical twin, at the initial time point and after they've aged. And what you see is the correlation between the hits is very tight within themselves, but not within each other. And we see the same thing for twin pair 9 and twin pair 6, all the way down the entire cohort, right? So everybody looks like themselves in the types of autoimmune patterns that they have across a decade, and they don't look like their identical twin. And that's because in the generation of each individual immune system, there's maybe 100 billion naive B cell clones that you start with, but it's impossible trillions of ways that those could have been made. And so the generation of each individual immune system and its potentiality for the way that it can make mistakes is completely individualized, even between identical twins. And I think this type of feature really hammers home why the serome is a place that people should be looking for, things that are not explicitly found in the genomics of individuals. And so we saw this same type of pattern using HuProt microarrays. So I'm just going to show the same result essentially pre-post using HuProt. And so these scatter plots are log transformed microarray data at either time point 1 or time point 2 for an identical twin pair. So this is one twin here. This is the person's identical twin over here. I'm not actually showing them between each other, but just trust me that the things that are coming up on the axis, they're very different from each other, just as on this plot. And the green spots are control-like proteins. So it's really these blue dots that matter. And so you see a cloud of maybe about 100 blue dots that's up here at this top end of the brighter stuff above 1,000 MFI on the signal in this person, same over here, and that very little changes, right? So it's maybe in a, if we were running replicate HuProts, this curve would be a little bit tighter, things would be pretty flat on the diagonal. There's a little bit of noise down here at the bottom end, but otherwise, these look like replicate samples. This looks a lot like if you just ran the same person twice. And these are people 10 to 15 years apart. So I think that observation alone is a really profound thing. And by the way, when you give things like nivolumab or ipilimumab, so you do anti-PD-1, anti-CTLA-4 checkpoint blockade therapy, a lot of the patients look like this pre-post. You can do some pretty profound things to the immune system that do not manipulate the serum. However, there are some patients, this is another twin pair. So this is one twin pair, here's another twin pair. This is a twin pair where something happened. So this person has a bunch of new responses. Their twin doesn't as much, but they do. They see a handful of additional things when they're older that they didn't see when they were younger. So what causes this? So we see things, so when we monitor cancer vaccine treatments pre-post, we'll see effects like this of new responses. But what causes it just in a healthy person? Did this person have surgery? Did they get a bad infection? We don't really know. But I think understanding and figuring out the origin of these types of effects is an important thing for our field and for Seromics itself going forward. So where do people actually use this stuff? So it can be in several different ways. So our pharma clients tend to come up to us with late stage clinical trials where they've got two different outcomes. So like the checkpoint blockade example that I used, or like with lupus, they'll have people that respond to a drug or don't, and they don't really know why. And there is really clear examples that we can help some of them with predictive autoantibody biomarkers, kind of independent of whether it's a mechanism, that help them determine whether people will respond or not respond to their therapy. Also, we have immuno-oncology clinical trials where they're really looking for new immune responses. They're looking for things like this. So wanting to look for that is something that cancer vaccine groups come to us with. If you're doing something else, like an immunotherapy that's not supposed to induce autoimmunity, say you're giving a antiviral vaccine, you do not want to see new autoimmunity coming up. And we haven't done a lot of very large trials with that. But for example, I ran a handful of examples early in the pandemic with people that had gotten either the Moderna or the Pfizer or the J&J vaccine. And comfortingly, in the pre-post examples that I looked at in that small cohort, we didn't see this. People look like themselves pre-post. And so it's a safety thing too. So you can observe for a lack of change in the autoantibody serum, and that can be a good thing. And also we do just pure biomarker hypothesis generation things. So looking at large cohorts, trying to figure out what things that might be autoantibody mechanisms that are associated with diseases. Additionally, people come to us not doing Seromics, but just using HuProt as a tool to help identify whether or not their proteins cross-react with things, or their drugs cross-react, or whether or not a drug does something. So kind of like I mentioned, sometimes these autoantibodies are function modifying. And so we actually have clients that come to us with libraries of isolated monoclonals where they know they do something, but they don't know what the target is. And so we do this type of target deconvolution work that I think is really, really powerful. Academic groups tend to come to us a little bit earlier in the profiling. So it's not so much late stage clinical trials, so much as that hypothesis generating exploratory stuff. And there's a lot of really interesting hypotheses that people have, and we're always excited to try to talk to people about their specific disease of interest, see if we can't come up with new leads. That's, to me, part of what's so exciting about Seromics is it's a place that a lot of people haven't looked yet. And every platform has its own advantages and disadvantages. And there are things that you can see on a HuProt array that have not been possible to be seen on anything else up until now. And so it's really exciting to go into those data types. And the rest of the uses are pretty similar to pharma. And so kind of with that, I think I'll stop there. I've talked about quite a few different topics at this point, and happy to take any questions.